Hello students, this is a module briefing for MM306 Bioinformatics. I am Dr. Yam Waikit from the Life Sciences Department of the School of Pharmacy at International Medical University. At the end of this briefing, you will be able to know the objectives and expected learning outcome of this module, know teaching staff for this module, describe teaching and learning activities, know types and breakdown of assessment and its deadlines. Introduces the field of bioinformatics and its application in life sciences. Bioinformatics refers to the generation and mining of computerized database of biological data. Genomics has to do with identifying the entire DNA code of not just one living thing, but comparing the complete DNA codes of many living things. Proteomics refers to new, fast, and efficient way to identify, compare, and study many proteins at one time. All of these tools are changing the way that researchers go about exploring the inner workings of cells. Bioinformatics lies at the interface between computer science, chemistry, biology, and technology. This module is divided into two parts, namely bioinformatics and structural bioinformatics. This module is an elective module for both conventional and ODL mode for Masters in Molecular Medicine. Similarly, for other modules in the program, passing mark for this module is 65%. This module aims to introduce current developments and applications of bioinformatics, provide practical instructions on the analysis of protein and DNA sequence using web-based tools, instruct on the various approaches used in structural-based drug design, instruct on the different methods used in elucidating the structure of biomolecules. At the completion of this module, students should be able to analyze sequences and protein ligand binding using in silico methods, evaluate and use the internet to acquire and manipulate complex biology information, apply bioinformatics tools in proteomics, functional genomics, and structural biology. Finally, to design structure-based drugs using various in silico approaches. Besides being the module coordinator, I will be conducting most of the teaching, learning, and assessment for this module. We also have Associate Professor Dr. Ganso Yi to cover the Biological Data Sequence Information Lecture and CAL session. We also have Dr. Lim Lei Ching to introduce protein and metabolic pathway. Finally, Dr. Wong Ying Pei will be involving in the assessment for presentation session. For ODL students, all lectures and discussion would be done in asynchronous mode. You will find all materials in the ODL portal or Teams. We also prepare online resources such as video, articles, or books for extra reading, which all of them is posted in the ODL portal. Although CAL sessions are not mandatory for you to attend, I would strongly encourage you to come for the synchronous sessions together with the conventional students. This is because CAL assignments would be based on CAL 2 and CAL 3, and you will be able to achieve the expected outcome of the assignment and also the module if you attend to the synchronous session. Assessment is consisting both formative and summative. Continuous assessment is done throughout the module via discussion, CAL sessions, or even in all of our asynchronous classes, pardon me. Summative assessment, on the other hand, would require submission in the ODL portal according to deadlines. CAL assignment 1 is based on CAL session 2, and that make up 35% of the total marks. While CAL assignment 2 is based on CAL session 3, which also make up to 35%. ODL students were required to record their presentation, upload it to OneDrive, and then share the video link with me. And finally, a 20% written report on the presentation topic would need to be submitted on the 31st of July. All requirements for each summative assessment are listed in the assignment instruction document that you can find in the ODL portal. That's all I have for module briefing. Please contact me via email or even Teams if you need any further clarification. Thank you and hope you will enjoy this module.